Here's a question. Which mixture makes an amide right away? An acid chloride with ammonia or a carboxylic acid with ammonia at room temperature? Lock in your answer. It's A. The instant reaction comes from acid chloride plus ammonia because acid chlorides are far more reactive. In this video, you'll learn what amides are, how to prepare them in the lab, and how they react, focusing on two key transformations, hydrolysis and reduction. Along the way, we'll build the model, work through examples, clear up a common misconception, and dive into mechanisms and edge cases. Let's start with the basics. An amide is a functional group where a carbonyl sits directly next to a nitrogen. They show up everywhere in biology, most notably as the peptide bonds that link proteins. So, what makes amides special? Resonance allows the nitrogen lone pair to delocalize into the carbonyl. This reduces the nitrogen's basicity, strengthens the carbon-nitrogen bond with partial double bond character, and makes the carbonyl less electrophilic. That's why amides are much less reactive than esters toward hydrolysis. That stability is vital in biology, but in the lab, we often need to make amides directly. The standard route is nucleophilic acyl substitution. First, convert a carboxylic acid into an acid chloride. Then let ammonia or an amine attack the carbonyl to yield an amide. For example, start with a carboxylic acid, treat it with thionyl chloride, and you get an acid chloride. Then, react that with 2 molecules of ammonia, and you get a primary amide plus ammonium chloride as a byproduct. Lastly, using a primary amine instead gives an N-monosubstituted amide, while a secondary amine yields an N-disubstituted amide. Here's a common mistake to avoid. Simply mixing a carboxylic acid with ammonia doesn't make amide under mild conditions. It just forms an ammonium carboxylate salt. To actually make an amide, the carboxylic acid must first be activated by converting it into an acid chloride or by using peptide coupling reagents in biochemistry. So far, we've defined amides, seen their resonance, and learned the standard lab prep. Want to turn those milestones into mastery? Tap the link to access the practice test, flashcards, and study guide, then jump right back into the video. Lock in what you learned and keep going. Now, let's look at their reactivity. Amides can undergo hydrolysis, breaking back into carboxylic acids. In acidic conditions, the carbonyl gets protonated, water attacks. And after a few proton transfers, the carbonyl reforms and ammonia leaves. The overall products are a carboxylic acid and an ammonium ion. Conversely, in basic conditions, hydroxide attacks instead. The intermediate collapses, releasing an amide ion that quickly grabs a proton to form ammonia. This gives a carboxylate salt, which only becomes the free acid after an acid workup. Beyond hydrolysis, amides can also be reduced. Unlike esters or acids, amides require a strong reducing agent. A strong hydride, like lithium aluminum hydride, removes the carbonyl completely, converting the amide into an amine. This is especially useful with lactams, cyclic amides, because reduction gives the corresponding cyclic amine. By contrast, sodium borohydride is usually too weak for this job. Before we practice, 2 quick reminders. Under basic hydrolysis, you only get the carboxylate until an acid workup restores the free acid. And compared to esters, amides hydrolyze much more slowly, often requiring heat and strong conditions. Now, try this quick challenge. Predict the products when an amide is treated under 3 different conditions. Pause here to think. Got your answers? Let's check. First, with hydronium ion and heat, you get a carboxylic acid and an ammonium salt. That's classic acid hydrolysis. Second, with sodium hydroxide and heat, then an acid workup, the base first gives a carboxylate salt and an amine or ammonia. if it's a primary amide. The acid step then restores the carboxylic acid. Third, with lithium aluminum hydride followed by water. The carbonyl is fully reduced, leaving an amine. Quick rewind. Amides are resonance-stabilized carbonyls. You can make them from acid chlorides with ammonia or amines. You can break them by hydrolysis under acid or base, and you can reduce them with lithium aluminum hydride to get amines.
Now that you've watched the video, let's see how much you remember. Head to the link in the description where you'll find Quizlet content made just for you.